Fueled by cancer, obesity, and cardiovascular deals, billion-dollar plus takeouts in biotech are on the rise. We'll take a closer look on the latest BioCentury This Week podcast. Plus, welcome to Ash Monday. Data have been pouring out of Orlando from the Hematology Society's annual meeting. We'll zero in on first-in-human in vivo CAR T data from Colonia. And more data. Another Praxis win provides much-needed progress in rare genetic epilepsies. And Selena got that data she was looking for. The full data set for Semaglutide presented at CTAD. Semaglutide was supposed to quench CNS inflammation. It didn't. Selena will dig into the details for you. And another tumultuous week at FDA. Steve will give his analysis on the back half of the podcast. I'm Jeff Cranmer, one of the executive editors here at BioCentury, and joining me today are my colleagues
Simon Fishburne, editor-in-chief.
Selena Koch, executive editor.
Lauren Martz, executive director of Biopharma Intelligence.
And Steve Usden, Washington editor.
It's back. BioCentury's indispensable JP Morgan Guide has returned. Visit jpmguide.com for the inside track on sideline events and receptions happening during the annual Biotech Kickoff in San Francisco. And hey, if you're hosting something special, use the online form that you'll find there to make sure your event appears in our guide. Again, that's jpmguide.com to make the most of your JPM 2026 week. There's a couple of big movers on data today. Structure Therapeutics, obesity companies, shares doubling in value as its once daily oral therapy leads to 11% weight loss, and Chimera rises 50% on strong biomarker results for its STAT6 degrader for moderate to severe atopic dermatitis. Both companies gaining billions in market cap. Stephen Hansen will have analysis of Structure's data on biocentury.com. So hey, let's kick things off. With a month remaining, this year is already tied for the most billion-dollar plus M&A deals of any year in the past 10. Selena, what's behind the trend?
Yeah, well, M&A has certainly been a bright spot. People keep citing. So we just do what we do here and wanted to quantify that. So by the end of November, so it's not yet full year data, so there could be more. There were $37 billion plus M&A deals announced this year, tying 2025 with 2021 for the biggest year in the last decade. And so we broke it down a couple ways. We broke it down by disease area, and we broke it down by the stage of the lead product of the company being acquired. And it probably won't surprise anybody that, companies with marketed products are the biggest set within that 37-12. But more interestingly, maybe there were 6 takeouts this year of $1 billion or more for companies in phase 1. That's up from 2 last year, 1 the year before, 0 in 2016, 2017. And even 3 preclinical companies were taken out for a high price tag. And this Lindsay's analysis coincided with a perspective piece written by our colleague, Paul Bananos, who had noticed a couple of these, big money takeouts of early stage companies, and had just posed the question, with pharma patent cliffs looming and a limited number of, say, mature assets available for M&A, are we going to start seeing buyers pay more for early stage companies? And, Lindsay's data suggests that maybe that's already starting to happen.
Yeah, I think it's really important because a lot of the focus when people talk about M&A is on consolidation of the big companies. And, we've been talking for a couple of years about looking at this rise of sort of billion-dollar plus deals where there's a lot of value in the, let's say $1 to $5 billion range and a lot of companies and assets. And, what you're talking about, Selena, is also regarding the patent cliff. Well, we know that a lot of the later stage assets have been picked off, right? So companies now need to go a little bit earlier in the value chain and, they're still willing to put considerable weight behind some of those bets that are obviously riskier. Obviously, we've got the J&J takeout of Halda for over $3 billion, which is a preclinical company. I think that raised a lot of eyebrows. But I think also maybe you could talk a little bit about the breakdown of therapeutic area that Lindsay's piece sort of laid out.
Yeah. Well, cancer remains king. If you look at the last decade's worth of these really large M&A deals, it's just the slope is just upward and to the right, with 2025 being the decade's highest number at 20 of them. For all the hotness that has been I&I, the last 3 years have been really steady for these large takeouts at like 7 to 10. And then the other growth area, of course, obesity and cardiovascular, seeing sort of year-over-year increases recently.
Yeah, well, I obviously for companies and in particular their investors, this is a good exit path given that the IPO market is still very constrained. So I guess we're going to imagine the beat of $1 billion plus M&A deals is probably going to carry on for a little while into 2026.
All right. Yeah. And that number, as Selena had said earlier, that's $9 billion plus M&A deals year to date for companies whose lead programs were in preclinical testing or phase 1 at the time the deal was announced. And that, too, the most in 10 years. Okay, Ash, unavoidable. If you have an email inbox, I'm sure a lot of Ash emails have landed in your lap. This week, Ash will deliver more first-in-human in vivo CAR T data that once again showcases the promise of the modality and justifies the growing pipeline. There's new data coming from Colonia Therapeutics. They show that 3 more patients have achieved MRD negativity after treatment with an in vivo CAR T cell therapy. My colleague Lauren tells me that the questions now include how long can the near perfect track record last, which technologies will prove safest and most effective, and how will these results translate to autoimmune diseases. Let's bring in Lauren now. Lauren, you going to answer all those questions?
Thanks, Jeff. I certainly don't have all of those answers, but with each clinical readout, I do think we get a little bit closer. As you mentioned, at a high level, what we've learned from the latest clinical readout for in vivo CAR T is that Colonia's KLN-1010, a lentiviral vector in vivo CAR T cell therapy targeting BCMA, is that it led to MRD negativity 1 month after treatment for all 3 heavily pretreated multiple myeloma patients evaluable in that study. We also know from the abstract that at least one of the patients remains MRD negative at 3 months, and we should learn more tomorrow when the full data are presented. I think another important thing about this readout is that this was profiled as a late breaker at ASH, which says a lot about how much excitement exists around in vivo CAR T cell therapies right now. We'll have a story tomorrow once the full data are reported. And for that, I've been looking into the pipeline of in vivo CAR T cell therapies. And right now, we were able to find nearly 40 programs. And this is what BioCentury's been able to identify. And that, I think, just reflects a ton of growth that's happened over the last few years.
So, Lauren, looking across the pipeline, what are the other ones that have reported data and, importantly, I guess, durability data?
Yeah, Selena, I think durability is the biggest question, and I'm not sure how much data we have to support that yet. When AstraZeneca bought ESO Biotech, we knew that they had efficacy data from 4 patients. I don't think there's much information about how durable the effects are. And in that case, again, it was a lentivirus targeting BCMA. I think that just remains a big unanswered question, and that will help, I think that will have a big impact on where this technology ultimately ends up being used as well. So durability seems to be very important when you're treating cancers. It may not be as important for autoimmune applications of in vivo CAR Ts. And the autoimmune applications are a bit farther behind, but I think we are starting to see some of the very early clinical proof of concept in those as well.
Okay, thank you for that, Lauren. Looking forward to seeing that one. Two other readouts, as I mentioned at the top, Praxis and Novo's Alzheimer's data for semaglutide. Let's start there. We discussed the readout on last week's podcast, data again at the Clinical Trials on Alzheimer Disease meeting. Selena, you were eager to see the full data. What did you learn?
Yeah. So the days leading up to the U.S. holiday of Thanksgiving, we just learned that semaglutide did not slow progression, but we didn't see any of the outcomes or the biomarkers. So this, in terms of a full data set, I mean, Novo had the data for like 2 weeks before the CTAD meeting. So it's top line. They didn't do a bunch of subgroup analyses or anything like that, but they showed the various clinical scales, the primary endpoint, CDR sum of boxes, pretty standard for Alzheimer's. And then there's a bunch of other standard clinical functional rating scales. And probably the most striking thing is that the curves over time of the treatment group and the placebo group are really just exactly on top of each other. There's no argument for a trend toward benefit or anything like that, really. It was a very clear failure. And as much as we hate to see failures, because they're not good for patients, at least the data were clear in that regard. The hypothesis, though, right, so you always have to ask yourself, okay, but did the drug do what it was supposed to do? And the hypothesis was that semaglutide was going to decrease peripheral inflammation, which was in turn going to decrease neuroinflammation, and that was going to be the mechanism by which it was going to help patients. You have to, measure all of those things to get a sense of whether or not it ameliorated peripheral inflammation. It measured high sensitivity C-reactive protein. That is what it measured. Novo measured in CVOT trials. It's cardiovascular outcomes trials, for example. And that was reduced about 30%, as was expected. But that was the only that was the extent of the characterization of peripheral inflammation. And there was just a big bucket of neuroinflammation markers. And apart from one small change on one of them, they were just not changed.
So that brings up the question that I had, which is, did you have to test a huge number of patients to learn that it didn't move the needle on these key biomarkers? Couldn't they have figured that out with, a dozen patients or 20 patients?
They absolutely could have done that work with fewer patients. Yes.
Right. And Selena did write that last week, but I also feel like Selena, they kind of underpowered this, because when you wrote that piece of they could have done it, what Steve is talking about, very important, how you're using your patients. But you pointed to the fact that they would learn a lot from this. And I'm sort of really wondering how much they did learn from this, because it was underpowered, and maybe power is the wrong word.
Power is the wrong word. We're only like 3,800 patients here.
So power is the wrong word, but it was not well designed, really, to get at the peripheral inflammation, because it only had this one very crude marker of C-reactive protein, right? Even though they had multiple markers in the brain.
Yeah.
If it had gotten into the brain, they'd have been full of data and like, it increases this marker, but not that marker. But they didn't really do a very rigorous job of understanding which part of the peripheral inflammation pathways it was activating. So how do you feel like in terms of the design and what you're able to walk away with?
I feel like the mechanistic piece, even though there were a lot of biomarkers, was a little underdeveloped. Yeah, I agree with that. Like, if I thought, okay, there's some crosstalk between the periphery and the central immune systems, I would want to know, what are the candidate pathways for mediating that crosstalk specifically? And that's not C-reactive protein, right? That's a general all-purpose tool and not thought to act directly at that interface of the blood-brain barrier. You might imagine doing a smaller, more focused study testing those specific hypotheses of how that communication might happen across the blood-brain barrier, which really wasn't done. Another idea for how the periphery can influence central inflammation is by the blood-brain barrier integrity kind of breaking down and peripheral immune cells getting in. They did have a few, two, I think, biomarkers of blood-brain barrier integrity, and those were unchanged by semaglutide.
Well, Selena, what I'm kind of hearing is that this molecule didn't really get into the brain very well, right?
Yeah, so that's an outstanding question. The thought is, based on the preclinical data and other data that's available, that it probably doesn't get into the brain in any appreciable amount. Novo did do a small study, like in conjunction with the phase 3 trials rather than before, which was also presented at CTAD. In 8 out of 10 people, there was a small amount of semaglutide in the cerebral spinal fluid after semaglutide treatment. I want to say maybe 1/10 of a nanogram per liter, something like that. So far less than you're seeing in the plasma, and strangely did not seem to correlate with the level in the plasma. So, and Peter Johansson, who talked about that, said very specifically, look, even if small amounts get into the CSF, it's a very different question whether or not any of that penetrates into the brain parenchyma in any appreciable amount.
Okay, so let me go back to Steve's question now, or Steve's point, which is you do this trial on 3,000 plus patients. You don't think you might do a little bit more work on whether the molecule gets in the brain?
I know. There are things that could have been done before jumping into a nearly 4,000 patient phase 3 trial, especially given the precious resource that patients are for trials.
Let's jump to some good news. Shares of Praxis Precision Medicine rose 31% on Friday after a phase 2-3 study of the company's sodium channel modulator for a couple of subtypes of developmental and epileptic encephalopathy was stopped early for efficacy. Stock is still moving up today. These came out of the American Epilepsy Society annual meeting. The full data came out Saturday. Lauren, this is a small molecule. Why is that significant? I know the seizures that they were evaluating are associated with a high risk of autism spectrum disorders. What did you learn in writing this story?
Yeah, I think the fact that this is a small molecule is one of the things that makes this data so exciting for Praxis and just for the field. This developmental and epileptic encephalopathy is specifically a genetic form caused by mutations in SCN2A and SCN8A. And here we have a small molecule that can treat specifically the genetic form of this disease. It's been a particularly difficult year to advance cell and gene therapies through the clinic. When you look at the pipeline for these types of DEEs, a lot of the development is focused in cell and gene therapies in particular. And so just given the commercial hurdles and some of the safety issues, I think it's just a good reminder that with the advances that we're seeing in small molecule drug development, there is an opportunity in some cases to design drugs that can treat these kinds of genetic disorders. And then the other thing that I think is really interesting here is that there's a genetic autism component involved here as well. So this is, a trial that's focused on epilepsy associated with these mutations. But SCN2A, for example, is also one of the most well-characterized genetic risk factors for autism. I think progress in helping restore function of the sodium channel could have an impact on autism. And this is definitely a hot area for drug development for different reasons right now.
Yeah, probably worth pointing out in this era of really tough funding for gene therapies, it's the safety issues we've seen this year, the continued commercial challenges. That modality piece is probably really important, and it's not the first. We saw Roche kind of prove this, that a small molecule can come into a space where there's antisense and gene therapy and kind of outcompete. That was Evrysdi in spinal muscular atrophy.
Yeah, but I think that's, both of these things are sort of converging on real actual steps within autism. Obviously, there's a lot of noise, political. That part of the program we'll get to after the break, I guess, Jeff, in terms of the politics around these things. But this is obviously about epilepsy, which is a feature of some autism spectrum disorders. But I think that there's a belief that there may also be benefits for some of the cognitive, let's say, features or other features of autism. And I think really what I'm trying to say is this is generally good news, not only for the epilepsy part of it, but also for the idea that with genetics and with these various approaches, there is progress being made, maybe slowly, but progress being made within autism. Is that how you look at it?
Yeah, Simone, that is how I see it. And I think it's also important to note that this EMBOLD study that was stopped early specifically looked at seizure reduction endpoints in the clinic for clinical efficacy. But Praxis Precision Medicine is also running a second registrational study that is looking at DEEs unrestricted, not limited to these specific mutations. And in that study, they are looking at different efficacy endpoints more broadly. Some of those are related to symptoms that may be more relevant to other aspects of autism spectrum disorders.
I think that's going to be key. And the other thing is if you treat very, very early, right? So these carry with them, these genetic defects, a risk of developing autism. So I wonder if we would see, in the real world data someday, a decrease in that risk.
All right, we're going to cut to a quick break. All of those stories you'll find on biocentury.com, biocenturypodcast.com. If you like what you're hearing, like and subscribe to BioCentury this week. We'll be back to talk about a wild week in Washington with our Washington editor, Steve Usden.
BioCentury This Week is brought to you by the 5th East-West Biopharma Summit in South Korea. An arc of innovation is emerging across Asia, and Western biopharma leaders are taking note, from cross-border deals to new co's. In March 2026, the 5th BioCentury Bay Helix East-West Biopharma Summit visits South Korea for the first time. Meet the biopharma leaders putting Korea innovation on the global map. Learn why Korea has become a clinical trial and manufacturing hub. Discover if Korea is the next hotspot for new co-formation. Plus meet biopharma innovators from India to Singapore to China and Japan. Register now at biocenturyeastwest.com.
We are back on the BioCentury This Week podcast. Let's get to Washington. Steve, I think you had a personal best in the number of reports that you filed last week. And where to begin?
Well, I think that when we left listeners to the podcast last week, Richard Pazder was still the director of the Center for Drug Evaluation and Research at FDA. There was some level of concern around a memo that Vinay Prasad, the director of CBER, had written about vaccine policy and a new framework for vaccine regulation. And things have, we've had another turn on the roller coaster, and this one is kind of a barrel roll. So first, Richard Pazder announced his retirement starting at the end of December. So he was on the job less than 3 weeks, and then he left. Almost every living former FDA commissioner signed on to a commentary that was published in the New England Journal of Medicine, warning that FDA's new vaccine policies constitute a public health crisis and expressing broader concerns about Commissioner Marty McCary's management of the agency. And then Tracy Beth Hoeg was named acting CDER director to replace Richard Pazder, who for the next few weeks has gone back to being head of the Oncology Center of Excellence. So you could hear the gasps from life sciences executives around the country and around the world when they heard that Richard Pazder was retiring as CDER director. And then you could hear the phones hitting the floor when they started getting the texts announcing that Tracy Beth Hoeg had been named acting CDER director. So the short form, she's viewed inside FDA and among regulated industry as unqualified for the job and an ideologue. She's likely to provoke a wave of resignations at FDA. That's what I'm hearing from people in the agency. And I think greater uncertainty for drug developers. Her appointment and Pazder's departure, or his imminent departure, I think are signs that drug reviews are going to be influenced by political pressures and ideological influences.
Just a minute before we get to Dr. Hoeg as the head of CDER, Steve, we've been running a survey, which is now closed, and we'll be getting that out this week. From the survey responses, here's a heads up to everybody, from the survey responses, as well as from a lot of personal interactions and conversations that we had at various meetings last week with biopharma BCs and biotech heads, there was really a feeling that Pazda's appointment was the biggest stabilizing force for FDA. You know, he was the grown up in the room kind of thing. Most of all, I think, is the stability. Now, his departure has certainly got a lot of people very worried about next year, even before they knew who the next CEDA director would be, right? What is there in terms of, have you got any good news, Steve? Is there any upside, calming things that you can tell people out there?
From FDA, there's no good news. I think there's news that people interpret as the possibility of good news, right? So, you know, we talked about it on the podcast before. There's this plausible mechanism pathway, which, leaving aside the name of it, has the potential to unlock the development of therapies for very rare conditions and bespoke therapies for very rare conditions. But it's far from certain that it's going to be implemented in a way that actually does that. But if you're looking for good news, you know, there's a potential there. There's also something else that happened last week, which was that one of the things that's been in the works at FDA for some time is an idea that Commissioner Marty McCary and CBER Director Vinay Prasad have of putting out a statement saying that the default assumption at FDA going forward will be that companies will have to perform a single pivotal clinical trial to support drug approval. They had been trying to get a statement saying that through HHS legal office for some time and hadn't been able to get final sign-off on it. Commissioner McCary discussed it, basically leaked the memo to a reporter from another publication last week. So it's been in the news quite a bit, but the press release still hasn't been released, so we don't know exactly what's in it. And what's more, we don't know how FDA might put it into practice. So you could look at that as good, you could look at it as bad. It's certainly going to create a great deal of uncertainty. You know, translating a press release into predictable policy is going to be challenging.
All right. So what are your expectations? What do we need to know about Dr. Hoag?
So she's an MD specializing in sports medicine. She has a PhD in epidemiology. She's never run a clinical trial. She's never reviewed a drug. She has no regulatory experience whatsoever. She rose to prominence as a COVID contrarian during the pandemic. She advised Florida Surgeon General Joseph Ladapo on anti-vaccine policies that have been heavily criticized by public health experts. She's played a leading role as FDA's representative to the ACIP, that's the CDC Advisory Committee on Immunization Practices, at which, in her capacity there, she's challenged the need for mandatory vaccination of children. She's called for changes to the vaccine schedule that will result in American children receiving fewer vaccines. As a senior advisor to FDA Commissioner McCary, Hoag's been closely involved with the Commissioner National Priority Voucher Program. She's close to CBER Director Vinay Prasad and helped write a memo, the one that I mentioned earlier, announcing new vaccine policies. Last week, every living FDA commissioner except one, Stephen Hahn, signed a commentary that was published by the New England Journal of Medicine that condemned that memo, saying it will have disastrous consequences for vaccine development and public health. People I know who have interacted with Hoag at FDA say she's scientifically curious. In meetings, she asks probing questions or wants to get into the details. But they also say that she's an advocate, not a regulator, that she starts with conclusions and cherry picks data to get to the desired response. And the FDA staff I've spoken with say Hoag has used that approach, and she's used observational data in ways that aren't scientifically appropriate to make assertions about drug safety. One example, she was in charge of a review of COVID vaccines that led to restrictions that were posed by longtime scientists and civil servants at FDA. Phil Krause, the former deputy head of vaccines at FDA, and Luciana Borio, the former acting FDA chief scientist, wrote a commentary that we published in BioCentury about the actions that Hoag took on COVID vaccines. And they wrote that shifting standards and late-stage demands for new data based on faulty scientific assumptions have eroded trust, delayed access to important tools, and discouraged developers from advancing vaccine innovation. I think we can expect that Hoag's going to move on MAHA priorities, restricting access to SSRIs and other psychiatric drugs for adolescents, taking action to reduce access to puberty blockers, supporting access to psychedelics, and other steps I think that are on the MAHA agenda. That's what we can expect that she's going to do.
Steve, I'm just curious, has Bill Cassidy made any comments about what's been happening at FDA?
So you're referring to Senator Cassidy, who's the chairman of the Senate Health, Education, and Labor Committee. He has made public statements about the ACIP. He's really kind of apoplectic about what ACIP has done in terms of reducing access of newborns or recommendations for newborns to get access to hepatitis B vaccines. It's a field that Cassidy knows well. He's a physician, and he treated a lot of hepatitis B patients, and he's extremely concerned about what ACIP has done. I haven't heard him say anything publicly about FDA.
Steve, well, I think, we would agree more with the orthodoxy on vaccines than, let's say, on the MAHA agenda. And I understand that Dr. Hogue has sort of had a lot to say about the COVID vaccines, also acknowledging that there are many people with different views about that. How relevant do you think her COVID vaccine position is going to be in her role as the head of CDER? Is there daylight between those?
Well, she doesn't regulate vaccines at CDER. CDER doesn't regulate vaccines. But I think that what it shows is how she thinks about things and the fact that she's willing and eager, really, to take actions and to take regulatory actions based on ideological principles rather than the scientific consensus. She's also acting, and this is consistent with what Marty Makary has been doing as FDA commissioner, in the absence of public advisory committees or other forums that would create transparency and accountability around FDA's actions on topics that affect public health and the health of individual patients, not to mention the ability of companies to develop drugs.
All right, Steve, a few other things are on the agenda in Washington. I don't know how much is going to get done. We've been talking a lot about the comeback of the Biosecure Act. Where does that stand?
So, the Biosecure Act has been included in the House version of the National Defense Authorization Act, the NDAA, which is a must-pass bill. It's a bill that's going to be passed very soon. It's almost certain that Biosecure 2.0 will be included in the bill. People may remember we've talked about this on the podcast. The new version of Biosecure differs from the version that was pending last year because rather than listing specific companies, it creates a process for the administration, right now the Trump administration, to determine whether companies are so-called biotechnology companies of concern. And for those companies, there would be restrictions on U.S. companies or companies that want to sell their products in the U.S. accessing their services. There's a 5-year grandfathering period, and there's also limits on the effectiveness of the sanctions, the effect of the sanctions on the ability of companies to sell drugs in the United States. A lot is going to depend on how the law is interpreted or implemented by the Trump administration. Another bill that's almost certain to be included in the NDAA is something called the Fight China Act, which imposes sanctions on certain Chinese entities and restricts or requires notification of U.S. investments in specified high-risk technologies in China. Basically, it establishes an outbound investment screening regime. It isn't directly targeted at biotech, and in fact, there are provisions in it that kind of provide a carve-out for certain research activities that are conducted in China. But nonetheless, it's likely to impact biotech companies. Again, a lot of it's going to depend on how the Trump administration interprets it, but some kinds of collaboration, some kinds of investments in companies in China that use advanced chipsets, for example, that are developed in China, or use advanced AI systems that are Chinese systems, could be subject to notification requirements or even have to disinvest in projects in China, depending on how the act is interpreted by the administration.
Okay, you can read all of Steve's stories, biocenturypodcast.com. We will be back next week. Don't forget to subscribe to BioCentury this week. And check out the latest from our sister podcast, the BioCentury Show. Selena sat down with Bill Hinshaw to talk precision medicine. Bill is CEO of 4 Biotherapeutics. Kendall Square Orchestra provides the music for BioCentury this week. The group connects science and technology professionals and other members of the greater Boston community to collaborate, innovate, and inspire through music while supporting causes related to healthcare and education.